Welcome back, everyone. Today I wanted to talk about a really excellent article that was recently published in the Journal of Invertebrate Pathology. I believe it was in last month's edition, so February 2025. And this is about a new parasite found in colonies of crickets. And the Journal of Invertebrate Pathology, if you aren't familiar with it, doesn't just deal with insects. It's primarily insects, but they also do a lot of things about shellfish and stuff like that, and all of their various diseases. Um, and it's a very, very well respected journal. It's a very old journal. But this paper has touched on something that I've spoken about before on this channel, usually in reference to news articles about the parasites of insects. And you really should not be eating insects, especially in reference to, oh, we need to start eating insects because of their lower greenhouse gas emission, and we need to use this as a meat alternative and all this other stuff. You really shouldn't be eating insects because they tend to be riddled with parasites, and specifically, they're riddled with parasites we know very little about because no one's really looking at it. And so this team from the UK and Denmark looked at common crickets that are mass reared. So these would be the crickets used for food and animal feed. And they found a new species of microsporidia, a new microsporidian pathogen, which, again, if you were not familiar with them, they're a unique form of fungus. And then they documented everything about this pathogen, gross pathology, microscopic structures, the effects on the hosts. It was just a fabulous paper. So I wanted to share it with you guys. So first, well, let's talk about some of the background information for people who aren't familiar. So this, the host system used in this was Gryllus bimaculatus and Gryllus assimilis. So this here is Gryllus bimaculatus. Very common crickets. You can probably pick these up from like the pet food store, stuff like that, if you're feeding lizards or turtles or what have you. And these are used extensively in animal feed with these kind of the advancement of insects as food for animals and people. And the problem with large breeding operations for insects is that they are extremely susceptible to parasite infestations, especially viruses and microsporidia. And microsporidia are a really cool set of organisms. They're probably my favorite set of organisms. I did a lot of research on these when I was doing my PhD, and they are a type of fungus, kind of basal in the evolutionary tree of the fungi, but they are single celled, similar to like yeasts, but they have a very unique cell structure, and this object inside them, this is normally kept coiled inside them, and it's called a polar filament, and they use it to inject themselves like a needle into their hosts. So this is how they're identified, and what they do quite a bit in this paper is describe the internal structure of this parasite when they're not talking about the effects on the hosts. But how these are different from other fungi is that they form a very thick spore which can persist in the environment. And then inside is the cell structure, like the nucleus and stuff. But they have this anchoring disc and this polar filament in blue that they keep coiled inside. And then this purple stuff is called the polariplast, which forms the cell membrane when they inject themselves. But basically, when they come in contact with a host, usually after a host has eaten them, this polar filament unwinds, it spirals out and injects the host cell. And then they have this vacuole in the back. This red part here is the vacuole, and it swells up. It takes in water, and it pushes the cell through this needle. So the cell will then go through this needle and inject itself into its host. And then it's actually a parasite that lives inside the cell of its host. And it's an energy parasite. And these sorts of parasites, these microsporidian energy parasites, are extremely common in arthropods and insects. And unfortunately, we know relatively little about them. Like, we know that a lot of them exist, but there's not a lot of people working on them. Um, and on top of it, they can affect vertebrates, they can affect mammals, they can affect people. So this is what they look like inside a host for the most part. This is the spore stage inside the host, not necessarily its active parasitic stage. But you can see it has a thick, this is a cross section, a transmission electron microscope image of a cross section. And you can see it has a thick spore wall. These little dots that you're seeing are the cross sections of the coil. So you're just seeing like the half cross section little nodes of the coil, and then they did the coloration here so you can see it. They're super cool parasites. They're very complex, but they tend to be very, very common in insects, and especially in insect rearing facilities, because they tend to get spread through a fecal oral route. So these have a tendency to infect the gut tissues of insects, and then when the insects defecate, when they spread their feces, and because you have a high density of insects in these rearing facilities, other insects will pick up the disease. Or frequently in these rearing facilities, the insects will cannibalize each other. So if an infected insect dies and it gets cannibalized, then all of the insects that ate it become infected. So it becomes a major issue for insect rearing. And actually, this was such a major issue. These were the reasons. This was one of the reasons that we figured out germ theory, that germs cause disease. Louis Pasteur worked on microsporidia in European silkworms, the silkworm industry, because they, in France, the silkworms kept dying off and they couldn't figure out what was killing them. And so he figured out that there was a parasite inside of them that was killing them, and it was a microsporidia, Nosema bombycis. Is that what it is? Nosema bombi? So, I think it's bombycis. The silkworm Nosema, and he figured out a way to selectively breed them to get rid of this disease out of the colonies. Anyway, so going back to this paper, now that we have some background information. So this paper, identification of Alboplastiflora grilli, which is a new species in food and feed culture systems. They did such a good job with this paper. Basically, they found this infestation, this parasite infected colony. I believe they came from Denmark. I don't quite remember, but they reared them out, and then they did a bunch of experiments on them. One, they did histology, so they dissected apart mostly infected females, although they also did it with males, and documented all of the tissues which were infected and took images of all the infections. They did electron microscope imaging of the individual parasites and the spores and all of the different life stages. And then they did purification, so they had pure spores, which they could then infect colonies. They could control an infection of colonies of these crickets. They did a DNA extraction and phylogeny and DNA sequencing for the whole genome, so that they could figure out what this was most closely related to as far as microsporidia goes. And then they did a series of controlled infections in which they fed colonies of both males and females these spores through the water gel that crickets eat. And then they did gross pathology on this. They did effects on fecundity. So how many eggs were the females laying, which is a known problem frequently for microsporidia, because microsporidia are energy parasites, and they frequently also infect the reproductive tissue of female insects. They will steal energy on a cellular level from the reproductive tissues, so that generally speaking, females tend to lay fewer eggs. And then they did tests on how much the insects were eating, infected versus uninfected, how much they were defecating, whether or not the disease spread faster in certain conditions, the effects on survival. They just did all sorts of stuff. So it was all really, really well done. So I wanted to show you what these infections look like. So this is the interior of one of the infected, I believe this was an infected male. And so you can see a bunch of the tissues in here. Actually, this was probably an infected female from the looks of it. I'm not entirely sure off the top of my head. But you have fat body and you have Malpighian tubules. So these are like the kidney, liver, sort of blood purification organs of insects. You have the gut wrapped around it here. And then you have these weird masses. And these weird masses are called zoonomas. And they're basically giant fungal tumors that form within the infected insects. And if you were to split this open, what you have are all of these spores. And what's interesting about this particular microsporidian is, and not all microsporidia do this, is it forms these packets of spores called sporophorous vesicles. So each of these packets contains between 8 and 16 spores, and they mature like this. So it's like the spores separate, boop, 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 and they keep dividing until they get between 8 and 16, and then they form this vesicle. And this is generally what is excreted from the host, but this vesicle will eventually split, and the spores will all kind of explode out to infect new hosts. But this particular microsporidian forms these giant zoonomas, which are not I don't want to say it's atypical, but it's not constant. This isn't something that you always see in microsporidia infections of insects. Frequently, the microsporidia will just infect either the Malpighian tubules or the gut lining, stuff like that. They don't always form these tumors, which is really interesting in this particular group. What you do see more is, if I had to guess, in this particular insect, these Malpighian tubules are particularly infected. They look a little swollen, and they're very, very opaque. So the opacity is probably from high density spore load within the tubules. And it's the same for some of these other tissues in here. So like this looks like more healthy fat body, whereas this looks like maybe more zoonomas. So then they did kind of microscopic histology of this, where you actually get individual tissues. So this is like the gut epithelium here, and this is massive infection within the gut of these microsporidia, and you can really see all of these spores forming within the gut tissue. Again, gut epithelium here, you have zoonoma formation within the gut, and you just have a lot more crazy stuff in here. So this is the cuticle, this is the exoskeleton of the insect, this is muscle tissue, and you have this massive infection of the muscle tissue. So let's zoom in here. You can see this is the striated muscle tissue here, and then you have massive zoonoma formation within the muscle tissue itself, which is kind of crazy. So this is not just like frequently with microsporidia, you'll have like single organ infections. So like nosema in honeybees tends to primarily infect the gut, and you'll find it in some other tissues, but it's not this massive sort of systemic infection. With this particular species, it seems to spread throughout the entire body. All of the tissues become infected. It's kind of crazy. I mean, it could indicate maybe that this is a relatively newer host-parasite interaction, although the hosts seem to be well adapted to this. But you have this like massive formation of infected tissue here. And then if you go to other systems, so this is, again, the cuticle. You have a little seta here. This is the cuticle. This looks like it might be a leg, a cross-section of a leg. And you have massive zoonoma formation within like the connective tissue. So where the muscles, it's almost like the tendons, where the muscles attach to the cuticle, you have this massive zoonoma formation. And what's cool about this, well, horrible for the insects, but cool to know about, is that eventually, because of this massive infestation of the connective tissue and of the muscle and stuff, the insects that have been heavily infected for most of their life at this point eventually become paralyzed, because this just eats through their muscle. And so they eventually starve to death, because this one is stealing energy, but it is also paralyzed them. So you have this really kind of vicious infection. But what's really cool about this is that even though this vicious infection exists, it doesn't seem to have a huge impact on the survival of the actual insects. So the insects tend to survive in just this very infected, heavily infected, depleted state to roughly the same age as healthy insects. But which is also kind of, I don't want to say typical, but generally microsporidia are a more insidious sort of parasite. They don't usually outright kill their hosts. they generally cause kind of a wasting disease in their hosts, including in humans when we become infected. Um, and so they don't necessarily, it's not like a virus or like these zombifying fungi that you see with like cordyceps and stuff, where it outright kills the host. Generally, what it does is causes them to a failure to thrive. It reduces their fecundity. It reduces their ability to feed, stuff like that. But what's crazy about this particular one is that in all these experiments that they did, it actually caused the especially the male insects to feed way more than usual. So this is it within the tissue. This is the final spore production. You can see the cross sections of that polar filament wrapping around the spore. But it caused anyway. It caused the males to feed more. So it had this really interesting effect called compensatory feeding, which these are energy parasites, and they're infecting the gut and everything else and the fat, and it's basically stealing the ATP that is generated from calories being burned. And so the insects have to, if they want to survive, they have to eat more so that they don't starve to death from this parasite stealing all of their energy. So the males ended up eating way more and producing an enormous amount of feces, which is how they measured this. With the females, there did seem to be initial effects of reducing egg production, although they did say within this paper, which I really like, is that they need to do more experiments with this because there did seem to be an inordinate amount of error in the measurements of the female egg laying. And so they wanted to do this again to make sure that this, because there is a trend here in the data where there is a reduction in eggs being laid by the females that are infected. But for whatever reason, with this 16 count density, there was a lot more error. So I'm not sure what happened there. But they did say there seems to be a trend, but they would need to redo this experiment. So what is the problem with this? So it doesn't necessarily cause outright death in the colony, although it does make them eat more. So you'd have to be feeding them a lot more if you were trying to produce these economically for like food. One of the issues is that this particular microsporidian is very closely related to human pathogens. So there is this concern that you could get the zoonosis effect of a jump from these crickets. If a human were to eat these infected crickets, that you could get a jump from that host to humans. And this is not an uncommon thing for microsporidia. Microsporidia frequently jump hosts. And the problem with microsporidia infection in people is, generally speaking, there's no cure. There are drugs available, but it's usually for suppression of the infections, not for cure of the infections. And the reason for this, why we can't cure them, is one, because they're a fungus, they're fairly closely related to animals, so to us. So a lot of the drugs that would hurt a fungus would also hurt you. It's kind of like chemotherapy, where it's like you want to kill the parasite before you kill the host, but that's really difficult to do with fungi, especially with microsporidia, because they live inside the host's cells. So it would have to be a drug that your cells would take up and therefore be poisoned by it. So most of the drugs available for microsporidia are for suppression of the infections, but once you are infected with them as a person, you will never be free of them. And they do cause this wasting effect. They tend to infect human digestive tracts. So you end up with like chronic diarrhea, failure to thrive, wasting diseases. And this is usually seen in people who are immunocompromised. So like cancer patients, AIDS patients, and transplant patients that are on immunosuppressant drugs. So this is a major concern for human health. Although it seems to be of minor concern to these insects, they seem to be adapted to survive despite these heavy infections. So it's an interesting situation. They had more ideas about what they were going to be looking at. I will link this paper in the description. It is open access, so everyone should be able to read it if you want. It's a medium length paper. It's about 18 pages of actual text, and then two or three pages of citations. But it's really, really interesting. The genetic stuff is kind of lost on me. I know what they're doing, and I've done genetic assays before and all this other stuff, but it's not, I haven't kept up with it. It's not my cup of tea. So if anyone is an expert on genetic sequencing and stuff like that, if you want to look at this and leave some comments on your thoughts, that would be appreciated. And I'll talk to you guys later.